So in this third lecture and the last lecture you're going to get from me on this topic, we're going to take what we've learned about immune cell metabolism and particularly lymphocyte immunometabolism, and see how the insights we discussed in the previous two lectures affect how the lymphocytes and immune cells work within nutrient adverse microenvironments. Like in particular, the best example would be the tumor microenvironment, but there are other microenvironments where the immune cells may find themselves that have quite metabolically difficult conditions. So you'll remember in the first lecture we discussed how in general metabolism is important for immune function. So, certain nutrients are key depending on the cell type. Some nutrients are more important than others. Glucose is particularly important for T cells, or for T cells and NK cells. Glutamine is very important for B cells. But you can imagine, based on the fact that metabolism is so important for immune function, if an immune cell finds itself in a nutrient restrictive environment, this may have implications for their ability to carry out their immune functions. We also discussed how all different immune cells have different metabolic configurations and therefore may be more or less sensitive to a particular deficit in the immune microenvironment, whether it be a decreased concentration of glucose or or fatty acids or other fuels. Different immune cells will be affected by that differently, depends on what type of fuels they rely on. And of course, we remember that on a basic level, metabolism is important for a range of things, including energy, biosynthesis, redox signaling, but also regulation. And we're going to talk about that a bit now. So regulating pathways, directly regulating signaling pathways to control immune function. And that's going to be important when we think about these immune cells operating in an immune microenvironment that is rather difficult in terms of supporting metabolism, whether that be due to inhibitory molecules that are produced by the tumor cells, or the low levels of nutrients due to competition by the tumor cells. Okay, so from the take-home message from the second lecture was that cellular metabolism is integral to lymphocyte responses. And we had slides such as these where we talked about how you could push differentiation of immune cells one way or the other, and you'll remember we talked about things like AMPK and mTOR being important, depending on whether they were turned on or off. You could push it either way. Glycolysis would be important. Well, obviously, if you've no glucose in the local microenvironment, this is going to affect glycolysis, and so therefore may affect differentiation of T cells. But equally, AMPK and mTOR are what we know as nutrient sensors. So their activity is acutely reliant on the availability of certain nutrients in the microenvironment. So they are also going to be affected, and we'll talk about that now in a minute. Same for CD4 differentiation. Again, there are similar molecules that are found to be really important. AMPK, mTOR, glycolysis is important. All things that are sensitive to what's available in the local microenvironment. So we start to think about this. We start to think, what's the implication then for how these cells operate at immunological sites where nutrient fuels might not be abundant present. So where glucose or glutamine may be in low levels, or where other metabolites that have the ability to inhibit their function are present in abundance or at high concentrations, like for instance cholesterol. And cholesterol metabolites like 25-hydroxycholesterol and 27-hydroxycholesterol, which of course we know to be potent inhibitors of the transcription factor SHREB, which we know is a key metabolic regulator in NK cells in particular, but also in T cells actually in more recent work. So let's start thinking about these different microenvironments. The tumor microenvironment, certainly we know that is a harsh microenvironment with low levels of certain nutrients and high levels of other metabolites that can be inhibitory towards immune cell function, but also sites of infection. these could also be, although we know a little less about the exact nature of the metabolic metabolites within the site of infection. We do know that, for instance, the cells like M1 macrophages can be abundantly present here, and they make high levels of 25 hydroxycholesterol. So there's lots of good reason to think that the immune microenvironment within sites of infection, whether it be virus infection or bacterial infection, is likely to contain metabolites that could be inhibitory, or to contain a deficit of nutrients like glucose, especially if you think about an infection like a Staph aureus infection, which is a bacteria whose primary fuel is glucose. You can imagine, as I think we discussed previously, that if bacteria is using all the glucose, the cells that are arriving to deal with that infection may have little glucose left to use. So I think we can think about metabolically adverse immune microenvironments to be both tumor microenvironments and potentially certain sites of infection. So the first part of this lecture, we're going to discuss competition for nutrients. So what's the consequence of there being insufficient nutrients for immune cells? Firstly, for their metabolic pathways, but then also for their signaling pathways. And we're going to talk on some interesting signaling pathways that are affected by nutrient levels and metabolite levels within the cell. And then we're going to finish up by talking about some of the work that we and others have done, looking about the actual impact of inhibition of metabolic metabolism or metabolic signaling on the cells in pathogenic environments. So looking at various diseases like obesity and cancer, looking at how the metabolism of the cells are altered in those situations. So as we know, or as I'm sure we probably know, is tumors are highly glycolytic, and they use a huge amount of glucose and have a very big appetite for glucose, and certain tumors also have a big appetite for glutamine. So you can imagine that levels of glucose and glutamine are likely to be low within the tumor microenvironment. We certainly know people have managed, they're not easy measurements to do, but people have managed to measure glucose levels within the extracellular space within the tumor, and they can be very low in certain circumstances. The measurements of glucose have been way more troublesome, but I think it seems logical that certain tumors will also have low levels of glutamine within the immune microenvironment. Again, as I said before, we know less about the nutrient levels within the site of infection, but they are likely to be affected here too, especially when you have a pathogen that also has a similar fuel preference to the immune cells, like for glucose. Also, it's also interesting to know that the other pathogens, like intracellular pathogens, actually change the cells that they infect to ramp up their metabolism. So viruses do this. So when viruses infect a cell, they will reprogram that cell to increase glucose uptake, to increase glycolysis, and also increase glutamine uptake. And this is for its own selfish purposes, because it's boosting the biosynthetic capacity of that cell, so that the cell can make more viruses. Basically, the viruses hijack the cell. But what it does mean is that at the site of a viral infection, you also have increased demand for glucose, because all the viral infected cells are taking up much more glucose than they would normally do, and this could lead to a sort of a lowering of the glucose concentration in the immediate microenvironment. Similar can be seen for some intracellular bacteria. They can also reprogram the cells that they infect to increase glucose uptake and glycolysis, again to support the growth of the bacteria rather than to support the growth of the cell, the host cell. And as I said, certain bacteria have a preference for glucose as a fuel store, and we know how fast bacteria divide and and so you can imagine at the site of infection you have billions and billions of bacteria all consuming glucose. So I think it's reasonable to assume that immune cells can encounter environments where glucose and or glutamine are at low levels. So what are the impacts that this would have on immune cells? So obviously there's a based on our first two lectures, this is going to affect the cells because it's going to affect their basal metabolic rates. So they won't be able to make as much energy. Potentially they have a lower biosynthetic capacity. We're going to talk about regulation now, because you may have different levels of metabolites in the cell, and that's what we're going to focus on today. And also you may have an inability to deal with reactive oxygen species because you haven't got the ability to make the NADPH to reduce your glutathione. So these are kind of some of the basic metabolic consequences, but we're also going to have consequences for signaling. Due to the altered activation of nutrient sensors, but also due to some of the changes in the amounts and abundance of some of these metabolites. So if you think of glucose, what are the pathways that are sensitive to glucose deprivation? Well, we know that both glucose and glutamine can be an important fuel for a cell, and if the levels of these nutrients are decreased, it can alter the ability of the cell to make ATP, and therefore you may get a decrease in ATP levels. And this kinase AMPK senses changes in the ratio of ATP and AMP, and when this ratio increases, AMPK becomes when this ratio decreases, basically you have more AMP than you do have ATP. This activates AMPK. AMPK then inhibits mTOR, and mTOR is a kinase that promotes all the anabolic processes that basically all the processes that consume energy. So basically, this is like an energy conservation switch. When you have low levels of ATP, you have to turn off all the processes that are using the ATP to make sure you don't run out. But of course, that means changes in the activity of these two important kinases that also have a role in controlling the fate of T cells and NK cells and other cells, in particular mTOR. So that's one consequence of glutamine or glucose deprivation, is activation of AMPK kinase, which is a glucose sensor. Well, in this case, it's an energy sensor. It can also be a more direct glucose sensor, because AMPK is also sensitive to this level of this glycolytic intermediate, fructose 1,6-bisphosphate. So if you've low levels of glucose, you get decreased flux to glycolysis. The levels of this metabolite will decrease, and therefore you get an activation of AMPK. So on multiple levels, AMPK is a really important glucose sensor. And by extension, mTOR is sensitive to glucose levels as well, because it is directly controlled by AMPK. So, as we said in B cells as well, we discussed the fact that glucose and glutamine are important fuels for energy for biosynthesis, but also these cells are particularly, glutamine is particularly important for these cells because it's required for correct glycosylation of antibodies, and the glycosylation in the Hinge region in the constant part of the antibody is absolutely essential for the biological activity of those antibodies, in allowing them to bind to the correct FC receptors on the various immune cells that it is communicating with. So this is the hexosamine pathway. It's involved both in just glycosylation of proteins, like the example of antibodies, but it can also do a thing called glutenacylation. Glutenacylation, you'll remember from if you were at my post translation modification lectures last year. Glutenacylation is kind of like a similar process to phosphorylation. It's an on-off process where you add a single glutenac, so it's a single sugar added to a serine or threonine, and then it can be removed by so OGT is a transferase that puts it there, and then you have an enzyme that removes it as well. And this is a way of controlling protein function, kind of similar to the way that phosphorylation controls immune function. And this is how glucose and glutamine can feed into the levels of c-MYC, and another important metabolic transcription factor in our cells, because the hexosamine pathway, which generates UDP glutenac, takes glucose and glutamine to generate this molecule. So you need a supply of both glucose and glutamine to generate UDP glutenac, which is a substrate for glutenacylation. This is taken by OGT, and you get glutenacylation of c-Myc or NF-kappa B is another example, or certainly others that are still to be determined. And this is an important consequence for c-Myc because threonine 58 is glutenacylated. If it's glutenacylated, that stabilizes Myc because it can no longer be phosphorylated, because it's a mutual it's competitive. So only the same residue can't be glutenacylated and phosphorylated. So if you get glutenacylation of Myc, you get stabilization of Myc, and you get driving of Myc-dependent processes, which include glycolysis and include increasing of mitochondrial synthesis. But if you have a deficit in glutenacylation, that same threonine 57 can instead be phosphorylated, and this phosphorylation marks c-Myc for degradation, and you actually get very quick degradation of c-Myc if you don't have this process going on. And also you can, if you block that phosphorylation, you can get stabilization of Myc. So it's a very acute process here where Myc is really acutely controlled by glutenacylation versus phosphorylation, and glucose levels and glutamine levels within the microenvironment are important in this process. So you can imagine decreased glucose glutamine may lead to decreased UDP glutenac in a particular cell, and decreased c-Myc, and certainly that's been shown to be the case. Most of the best work on this has been done on CD8 T cells. So that's another consequence of low levels of glucose or glutamine. There's also this lovely story that came out a good few years ago now, that showed that one of the glycolytic intermediates, phosphenyl pyruvate, which is the last intermediate before you get the pyruvate, the terminal metabolite of glycolysis, and this seems to play an important role in controlling calcium signaling within the cell. And they did this in T cells. And calcium, of course, is a very important signal during activation of T cells, because it controls NFAT activation. And you'll know that NFAT is a really important transcription factor in activated T cells for driving a whole range of the driving the expression of a whole range of the key molecules required for efficient T cell activation. Of course, NFAT stands for nuclear factor of activated T cells. And one of the key processes that must happen is that NFAT must be dephosphorylated by this phosphatase, so that it can relocalize to the nucleus, where it can then drive the expression of its target genes. And calcineurin is sensitive to calcium levels. So basically, you get activation of T cells, you get an release of calcium from the ER here, and then you get activation of calcineurin, NFAT translocation. But as with everything else, when you activate a signal, you always have to turn it off again, and this is where this transport becomes important. So after you have activated the pathway, your activated T cells, that's you start to reuptake the calcium back into the ER, and that will ultimately lead to an inactivation of the signal that's driving calcineurin NFAT signaling. And so that's a way of turning off NFAT signaling when you're finished activation. Now, phosphenyl pyruvate is important because it controls the activity of SERCA, which is this uptake transporter. So if you have high levels of phosphenyl pyruvate, you manage to inhibit the SERCA transporters, which means that the calcium remains in the cytosol for longer. You get robust and prolonged NFAT activation that's required for efficient T cell activation. If you're in a glucose deficient microenvironment, low levels of glucose mean that you get decreased levels of all the metabolites throughout glycolysis, including phosphenyl pyruvate. And under these circumstances, following TCR activation and T cell activation, you still get calcium released into the cytosol, but it's taken up much quicker back into the ER, which means you only get transient and less robust NFAT activation, which ultimately leads to inefficient and an inability for the T cells to activate properly. Really nice paper, and this is the paper here from in 2015, which you can go and have a look at if you want to know a bit more. But there's an example actual metabolite directly controlling one of the fundamental pathways for controlling T cell activation. So and there are more examples. You're going to hear more examples of metabolites controlling key pathways are required for myeloid cell activation when Luke gives you lectures on macrophages in particular. Is there any other ways? So we're still on glucose, well, and glycolysis. Well, there certainly was this lovely, a nice paper going back a good few years as well, showing that the glycolytic enzyme GAPDH can have multiple roles in the cell, so it can act as a glycolytic enzyme, as we've discussed in previous lectures. But it can also bind to RNA, so it can bind to 3' UTR of messenger RNA and inhibit the translation of the RNA. So if GAPDH is bound to mRNA, it will reduce the amount of protein that's made from that RNA, essentially. And we know that it binds to the mRNA for two important cytokines that are produced in activated T cells, IL-2 and in interferon gamma. So here's an example where glycolytic enzyme has the potential to control the expression of these two cytokines. And the idea here is that when glycolysis is fully engaged, GAPDH is engaged in glycolysis and it's not repressing mRNA translation. But when you get reduced levels of glycolysis, GAPDH can now engage in this sort of this other role. It's moonlighting function as an inhibitor of translation of these particular transcripts. Now, we're still waiting for a sort of follow-up study, and I think more will emerge in this area as to other genes that may be controlled by GAPDH and potentially in other cells as well. But it's also another very sort of unique and novel mechanism through which glycolysis and glucose can directly control the function of lymphocytes. In this case, it was done on CD4 T cells and some of the key cytokines that they produce. So it's metabolism directly controlling the immune function, which is really quite interesting. So what about glutamine? Any other implications of glutamine deprivation? Well, we know in our NK cells that we work on, and also in T cells, that glutamine, one of the main reasons why glutamine is really important for those cells is because it supports the uptake of other amino acids through SLC75. So SLC75 is a large neutral amino acid transporter that's really important in T cells and NK cells, maybe less so in other immune cells. It takes up amino acids, a range of amino acids, including leucine, but it's a antiporter. So for amino acids to be taken in, you must have an amino acid that's exported as well. And the amino acid that has the lowest KD for export is actually glutamine. So glutamine is really important for this to be exported to allow these amino acids in. Now, of course, leucine is particularly important because leucine is known to be it was found to be one of the first regulators of mTOR complex, because mTOR was initially one of the first pathways that was found to regulate mTOR was amino acids, in particular a couple of amino acids, of which leucine is one of them. So leucine binds to this cestrin 2, which we know is the leucine sensor, which then goes on to control mTOR1 function. So if you take away leucine, you lose mTOR function within minutes. So that's another reason why glutamine is important to allow this. And we also know that if you take away glutamine in T cells and NK cells, you also lose mTOR signaling within minutes because it's important for the uptake of leucine. Just to say here, there's another amino acid sensor that's important for mTOR signaling in response to arginine. This is called Castor. So arginine transporters are also important. And there are certain circumstances which we'll discuss where arginine can be depleted in the immune microenvironment, which might have an effect on mTOR signaling. And it's depleted. The reasons it's depleted are more to do with blocking some of the antimicrobial functions of myeloid cells like neutrophils and monocytes. Okay, so let's expand this out and start thinking about, on one level, the signaling within the immune cell of interest, and how cells in the local microenvironment, so its neighboring cells, can affect those signalings due to what they can do to the levels of nutrients, amino acids, glucose, etc., in the microenvironment. So we're talking about, in the first instance, competitive uptake and use of nutrients and how this impacts other cells in the local microenvironment. So we've already discussed about glucose, how if you have a cell like a tumor, so you may have tumor infiltrating lymphocytes here into an environment where there's loads of these tumor cells that are taking up huge amounts of glucose because that's what they do, leaving a situation where the levels of the glucose available in the microenvironment are quite low. How does that impact signaling within these tumor infiltrating lymphocytes, for instance, or any immune cell? Well, we know that glucose controls the level of NFAT signaling. We know that it can link to to MYC signaling. It can be sensed by AMPK and mTOR. So here we have some really important signaling molecules that are sensitive to the levels of glucose. NFAT, one of the key, particularly important in T cells. c-MYC, we know that c-MYC is very important in T cells and NK cells, mTOR as well. It seems to be important in most immune cells in some way in controlling their function. So here we have a situation where now glucose can control some of the really important signaling pathways within immune cells. Glutamine, then, we've also discussed. It also controls a number of pathways. Glutamine can potentially affect MYC stabilization. It can affect mTOR1, particularly these ones. Glutamine can affect mTOR signaling, and as can a few other amino acids like arginine and leucine. So you potentially could have competitive uptake of leucine, but we know little about leucine levels within immune microenvironments. But arginine is interesting because we know that there are processes that can actively deplete arginine in the immune microenvironment, such as enzymes like arginase, which can be aberrantly expressed in certain pathological microenvironments. So arginase will convert arginine into ornithine, and this can be a way to deplete arginine from the microenvironment, which would have implications for arginine dependent processes, which includes controlling the signaling molecule mTOR. Another example of enzymatic depletion of a nutrient is IDO, which depletes levels of tryptophan in the tumor microenvironment, converts that into kynurenine. And this is a particularly interesting pathway because depletion of tryptophan has implications for the immune cells, but kynurenine is also an inhibitory molecule that can also control immune cells itself. It's kynurenine and its metabolites. So tryptophan is able to activate another kinase, which we haven't discussed much yet. Another amino acid sensor kinase called GCN2, which has implications for immune cells. This is more of a general amino acid sensor, so it actually senses. I'm not sure if I'm going to talk about this in the next slide. No, I'm not. It actually senses the levels of uncharged tRNA. So if your general pool of amino acids becomes depleted, you get uncharged tRNA, as in a tRNA without an amino acid attached to it, and that's what activates GCN2. So quite a general amino acid sensor. So there's lots of pathways that can be sensitive to nutrients, and as I said, kynurenine is also a molecule that can actually, and I think it's on one of the next slides, that can actually control another pathway which can control immune function too. So as you can see from this slide, there are lots of reasons why nutrients may not be abundantly available to an immune cell that arrives at a particular immune microenvironment, and this has implications directly for metabolism, but also for some key signaling pathways. I do the slide here. Look, so here is just a brief slide describing how GCN2 becomes activated. So you have your general pool of amino acids that is fed from, you can make amino acids from autophagy. These amino acids will be fueling protein synthesis through translation, and the pool will also be topped up by uptake from external environment. And we know that once the pool of amino acids becomes depleted, you get charged tRNA that will lead to activation. So uncharged tRNA leads into GCN2 activation. And it's been seen that just this enzyme itself and depletion of tryptophan in certain circumstances can be sufficient to drive the activation of GCN2 in immune cells in that environment. So that's GCN2, and kynurenine can also affect pathways, because either kynurenine or some metabolites generated from the metabolism of kynurenine can act as ligands for this transcription factor AHR, which also has roles in particular described in CD4 T cells. Now, while these are all sort of your classic nutrients, we mustn't forget oxygen. You know, oxygen can also become limiting within the tumor microenvironment and other immune microenvironments within the gut, for instance. And of course, HIF1 alpha is a key oxygen sensor. When levels of oxygen decrease, you get an increase in HIF1 alpha. And we know HIF1 alpha is also important in controlling immune functions. There's a range of literature on that, and in quite a diverse range of immune cells. So you have here lots of sensors of lots of different things, ranging from oxygen to glucose to glutamine to amino acids. All these things have the potential to be changed within an immune microenvironment. So, for instance, here's a couple of examples. We know that within the tumor microenvironment, arginine can be consumed by enzymes. So, for instance, INOS is another enzyme that uses arginine to make nitric oxide. You also get expression of arginase by tumor microenvironment associated cells. So we know that arginine can be aberrantly expressed in fibroblasts in tumors or in tumor associated macrophages, and these cells will be depleting the levels of arginine available for infiltrating T cells, for instance. And we know that, as per the previous slide, that arginine is important for mTOR activity, but actually arginine is also just important for the basic metabolism of T cells and NK cells, and we know that arginine deficiency affects these cells' ability to show anti-tumor effects or anti-tumor functions. Tryptophan, there's also evidence that IDO can be highly expressed by tumor cells or some tumor-associated cells like tolerogenic DCs. So these are dendritic cells, so have pro-tumor functions. And we know that IDO can inhibit T cells and NK cells, and the mechanism is a combination of tryptophan depletion, so IDO using up all the tryptophan, and also the production of this metabolite that we've talked about, kynurenine. That affects their function, at least in part due to this aryl hydrocarbon receptor. So that's modulation of tryptophan levels, active modulation of tryptophan levels within tumor microenvironment. If we look at sites of infection, there are other examples where these metabolites can be altered in expression, and this tends to happen when a pathogen has developed a strategy of immune evasion that involves depleting some of these nutrients. So, for instance, Helicobacter pylori bacteria express arginase, and this is beneficial to these bacteria because it depletes the local microenvironment of arginine, and in the absence of this amino acids, the macrophages that are coming to deal with this infection have an inability to make nitric oxide. So one of the antimicrobial tools that macrophages have in their tool set is the production of nitric oxide, and in the absence of arginine, they now fail to make the same amount of nitric oxide, which is beneficial towards the bacteria. So here's another example. This is the intracellular parasite Leishmania major, which expresses arginase to deplete the host cell of arginine. Again, this impairs the ability of the host cell to make nitric oxide, which is one of the key antimicrobial mechanisms that it has. So again, a mechanism of evasion of the immune response. Now, in terms of the role played by IDO in the context of infection, we know less than we do about IDO in the tumor microenvironment, but certainly it is implicated in the growth inhibition of certain bacteria, parasites, and viruses. It's expressed in cells in response to a range of inflammatory stimuli, including those associated with infection like interferon alpha and TGF TNF alpha. So and you know it's likely that this enzyme is going to have an impact upon the metabolic environment in the context of infection. Bacteria. Okay, so taking the whole picture, we have discussed how there are situations where certain nutrients may become depleted in the immune microenvironment, either through competitive use by cells, the other cells like tumor cells in the microenvironment, or through active enzymatic depletion of those nutrients, which is the case for arginine and tryptophan. And these have effects on key signaling pathways like aryl hydrocarbon receptor, which senses kynurenine, GCN2, which senses amino acids, and mTORC1, which is its activity is altered by glucose, glutamine, arginine, leucine, and c-MYC, which is also sensitive to glucose and amino acids. So what are the immunological outcomes of all of these signaling changes? Well, we know that aryl hydrocarbon receptor signaling can promote Tregs, so that's interesting in the context of tumors where Tregs are a negative prognostic factor for tumor survival. So we don't want Tregs in tumor, but potentially our AHR or kynurenine production and AHR may lead to that to high levels of Tregs in certain situations like that. We know that GCN2 signaling is linked to both Tregs, Th17s, CD8 T cells, and DCs. So it promotes Tregs and inhibits Th17 differentiation, inhibits CD8 functions, whereas it can increase certain functions of dendritic cells. So again, clearly a nutrient able to affect signaling to alter immune differentiation and function. mTOR has described roles in most immune cells at some level, but it's particularly known to promote Treg differentiation, and you need lower levels. It's particularly known that turning off mTORC1 will lead to Treg differentiation and inhibit effective T cells, inhibit NK cells, although have some, but also promote some of the pro-inflammatory functions of dendritic cells. And MYC, again, we know MYC is very important in T cell differentiation and function and NK cells. So there are immunological consequences for altering nutrient levels in the immune microenvironment. So this is the kind of final picture we have here. Of course, I would be remiss not to add one last metabolite that is actually produced by tumor cells and other glycolytic cells, and that's lactate. So obviously we know that in the context of tumors, they are really engaged in this aerobic glycolysis. This is what Warburg saw those 80-something years ago. And the consequence of that is a production of lots of lactate. And when lactate is exported, it's exported alongside a proton, so you get acidification of the local microenvironment. And the tumor microenvironment can be quite acidic. And we now know that also has negative function or negative implications for immune tumor infiltrating immune cells, including NK cells and T cells, the two cells that can directly recognize and kill tumor cells. So that's another important metabolite that controls and impacts upon the immune response to tumors in this case. So some nice studies that showed you the importance of lactate production in altering the immune microenvironment. Here's an example in mice where they looked at lactate production, or at cells that had differential levels of LDHA, and they found that in the lack of it would decrease lactic LDHA. So this is the enzyme that makes lactate dehydrogenase A. This resulted in smaller tumors because there was less acidification of the the tumor microenvironment, meaning that the NK cells and the T cells had a better function, better ability. So, and this is measured as, you know, CD25 is a marker of activation of these cells. Interferon gamma is a key effector molecule produced by these cells, and so better immunosurveillance and decreased tumor growth. Whereas when you have tumor cells that overexpress and have high levels of lactate dehydrogenase, they make lots of lactate. You get more acidification of the tumor microenvironment, which inhibits the function of these NK cells and T cells. They make less interferon gamma, show less activation. You get increased tumor growth, and they showed that in this paper, they're focusing on CD8 T cells. They showed that, you know, the pH was really affecting things like simple signal pathways like NFAT signaling. So a very simple study that showed you how altering the amounts of lactate produced can affect the growth of the tumor. And of course, then there's also this lovely study published a couple of years ago from our own Clina Farley's group, where they showed that the acidic tumor microenvironment in patients who have metastatic colon cancer in the liver was affecting the liver NK cells, impairing their functions because it was driving a reduction in the pH within the NK cell, leading to NK cell death. So another very nice study showing that, you know, the cells that are potentially going to be the ones exerting the anti-tumor function are inhibited by some of the metabolic characteristics of the tumor microenvironment. In this case, high levels of lactate, low levels of pH can actually cause apoptosis of the resident liver NK cells. Okay, so let's go on to the second part, where we're going to briefly talk about a few diseases. So, as I've said, you know, in general, these key signaling pathways can be sensed by key nutrients that can be altered in the tumor microenvironment. But also some of the immunosuppressive cytokines that are associated with cancer, for instance, also have the ability to inhibit mTOR. TGF-beta is an important one that's elevated in a lot of cancers, such as breast cancer. And TGF-beta can also directly, although the mechanism is not quite clear, it can also seems to be able to directly inhibit OXPHOS mitochondrial respiration. But there are also other metabolites that can affect these key metabolic regulators, such as oxysterols. We've discussed as we went through. They are direct inhibitors of SHREB processing and SHREB activation. But also lipids. So fatty acids have been linked to metabolic dysfunction, in particular in NK cells, through the activation of PPAR signaling. So, for instance, we have done, I'm going to show some data for us in a second, of how this is true in obesity, but also there's a recent paper showing in a mouse model of B lymphoma that the NK cells in those experimental mice were loaded loaded up with lipids, and that was inhibiting the NK cell function. So basically, they were packed full of too much lipid, which has a negative impact on the NK cell metabolism and function. So this is the study we did with some of our collaborators. You recognize Lydia Lynch, Donald Lachey, and Andy Hogan, who is now in Maynooth. We all collaborated together on looking at natural killer cell function in obese adults and children. And it's very clear, we've known for a while now that NK cells in obese patients are severely impaired in their function. And what we did is we took some of these cells and stuck them into our metabolic analysis. So the machine we use is called a CRS extracellular flux analyzer, and looked at their ability to undergo glycolysis here on the x-axis, measured by extracellular acidification rate, and OXPHOS here on the y-axis, measured by oxygen consumption rate. So you probably heard about this process from Richie Porter earlier in the year. And if you take NK cells from the blood of lean patients or lean individuals, some more patients, you can see that when you activate them with cytokine, you get this really nice metabolic response where you get increases in both glycolysis and OXPHOS. But if you take NK cells purified from the peripheral blood of obese patients, you can see that there's absolutely no metabolic response whatsoever in response to cytokine production or cytokine assimilation. So it's very clear that metabolites can affect lymphocyte function. It's very clear that lymphocytes are dysfunctional in a range of conditions, including tumors, including cancer, and, for instance, chronic viral infection. We also get dysfunction of T cells and NK cells. And one of the common themes of this, which I kind of suggested to earlier, is it can be metabolic health in terms of the mitochondria. Chronic stimulation can lead to mitochondrial dysfunction. Alterations in metabolism can also lead to changes in mitochondrial structure and function. So we discussed earlier in an early lecture it's important of this sort of dynamic process where mitochondria can fuse to generate these elongated networks, and they can also undergo fission to generate smaller mitochondria. And this is an important process. If you have any mitochondrial dysfunction, you can pinch off the dysfunctional part of the mitochondria, and then they can be removed by mitophagy. But something we see in some chronic diseases, a common theme we see in chronic diseases, is mitochondrial dysfunction, both in terms of the amount of mitochondria in the cells and their structure. So here's a study from Nature Immunology last year, 2 years ago now, that looked at NK cells in the periphery, liver NK cells, and then tumor infiltrating NK cells in a hepatic carcinoma model. Actually, they're carcinoma patients. So and here you can see some electron micrographs looking at the mitochondria here, and you can see here up here you can see the mitochondria are quite long. There's quite a few of them, so they're definitely elongated here, as you can see here as well. Whereas if you look in the tumor infiltrating NK cells, the mitochondria are very small, very few of them. You can see here again, they're not forming any sort of elongated networks to speak of. And here you can see the zoomed in photo micrographs, and you can see here is a nice long mitochondria. You can actually, if you squint, you can almost see the cristae as well. Whereas you can see here you have these very just oval mitochondria. I think if you got a better image, you would see that, and there's other studies to show this, that even the cristae are a little more disorganized, whereas here you can see that the cristae are actually quite regular and organized. And they also looked at this by sort of fluorescence imaging here using a mitochondrial dye. You don't get the same resolution, but it's still very clear, and shows you that the mass of the mitochondria for these NK cells is much more than from the NK cells that are infiltrating into the tumor in these patients. Similar has been seen for T cells. So this is a work by Greg Delgoff in Philadelphia, and he showed also that in tumor infiltrating CD8 T cells there were differences in the mitochondrial mass and structure and function. Here you can see in the tumor infiltrating T cell these small little mitochondria compared to this long elongated mitochondria in the control CD8 T cell, and you can see here the really nice structure of these cristae forming these invaginations here in the mitochondria, and this is sort of the structure that you expect from a cell that's efficiently engaging and able to efficiently engage in oxyphosphate energy metabolism. Whereas here you can see smaller mitochondria and less ordered mitochondrial cristae. There's a similar idea in chronic viral infections. So we know that T cells and NK cells can be found to be exhausted during chronic viral infection. There's a couple of nice papers on that in here, and in general what they see is that these exhausted T cells in chronic viral infection have altered levels of metabolism, sometimes even increased glucose uptake. But they also tend to have really dysfunctional mitochondria in terms of mass, and again in mitochondrial structure. Okay, so I think the take-home message from this lecture is that metabolically restrictive microenvironments can have a significant impact upon the immune upon the functions of immune cells. And we kind of focus a bit more on T cells, on NK cells, and in the context of tumors. But I think this is going to be a common theme throughout a wide range of immune cells and different immune microenvironments. And I think the site of infection could also be an important microenvironment that is difficult to maintain a proper immune function over time, and chronic infection could lead to really pronounced immune dysfunction over time. So I hope these 3 lectures have made sense to you. I hope that you now have a good understanding of why metabolism is important for immune cells, why different immune cells can have very different immune metabolism, and how these metabolic pathways and nutrient requirements will have an impact for how these cells are affected by certain microenvironments. So, as I said, there's some nice reading material here that you can dip in and out of if you want to find out more information on NK cell metabolism, for instance, down here, or just general immunometabolism or T cell metabolism. There's a nice review by the Pierce group. Okay, so listen, I hope you enjoyed these 3 lectures, and that you enjoy Luke's lectures on immunometabolism too. Okay, thank you.